Synthetic biology, which is defined as the application of engineering principles to the redesign of biological systems to perform novel functions, is a fast-growing field of science. This is commonly attributed to the advances in DNA sequencing, synthesis, and gene editing tools available, allowing synthetic biologists to program cells at will. Most of the time, synthetic biology is applied to living cells such as E. coli and yeast, allowing them to turn into anything ranging from microbial sensors to microbial factories. However, it is well known now that living cells are very complex and contain barriers such as the cell membrane, which either prevents or hinder the engineering of these cells. Due to this, synthetic biologists face the challenges of difficulty to standardize, unwieldy complexity, incompatibility, and variability when engineering cells. These challenges were presented in an article titled 5 Hard Truths for Synthetic Biology by Roberta Quirk. Fortunately, an emerging interdisciplinary approach is being investigated to overcome these challenges: cell-free synthetic biology. Cell-free synthetic biology is, as the name suggests, synthetic biology but applied to biological machinery without the use of living cells. The machinery parts such as RNA polymerase, ribosomes, the DNA templates such as a plasmid, amino acids, and tRNAs are combined in an open environment containing buffers and salts to mimic the cellular environment. However, due to the lack of a cell membrane, this environment can be altered to however someone may please. Consequently, cell-free synthetic biology has a range of both research and industrial applications, such as protein engineering and synthesis, metabolic engineering, and even the detection of Zika virus. There are two main types of cell-free systems. One is extract-based, meaning the system is composed of a crude extract obtained from organisms such as E. coli, which contain the transcription and translation machinery, DNA templates, nucleotides, amino acids, cofactors, and salts. The second is purified enzyme-based, which utilizes purified enzymes and coenzymes to create synthetic enzymatic pathways. Protein engineering is a valuable technique for both research and industrial applications. However, the process is reliant on cell-based approaches, which presents a number of challenges. Firstly, insoluble expression causes a reliance on cell lysis methods for purification, and secondly, there is an inability to control the rate of transcription and translation. Cell-free protein synthesis can overcome these challenges due to its high modularity and ability to optimize the conditions of the environment based on what type of protein is being synthesized. For example, a study published in 2008 utilized an E. coli-based cell-free protein expression system, plus the addition of nanolipoprotein particles, or NLPs, to express and purify a number of membrane proteins, which are notoriously hard to express and purify in cell-based approaches. This is due to their insolubility from their hydrophobic nature, frequently resulting in protein aggregation and cell toxicity. The NLPs self-assemble into disc-like particles that are water-soluble and can be used to encapsulate the expressed membrane proteins, which greatly simplifies the purification of these proteins. Metabolic engineering is commonly employed to genetically engineer microbes, making them living chemical factories that synthesize a range of products such as biochemicals, biofuels, and even therapeutics. However, one issue with this is even though these microbes are chemical factories, they are still required to spare resources to cell growth. Cell-free synthetic biology offers an alternative solution to metabolic engineering as it diminishes the concerns of cell growth and complicated metabolism, and enables many biomanufacturing advantages, such as a fast synthesis rate and a high yield. A study published in 2011 details a scalable cell-free production of an active protein known as granulocyte macrophage colony stimulating factor, or GM-CSF, which is a cytokine that functions as a white blood cell growth factor and has therapeutic applications, such as it being used to accelerate white blood cell recovery following a bone marrow transplantation. This study effectively demonstrated how cell-free synthetic biology can be applied to metabolic engineering to produce a desired product in a scalable quantity. Zika virus is transmitted through mosquitoes, similar to malaria and dengue fever, and may cause serious negative health effects. It currently has no specific treatment or vaccine, hence a significant amount of research is occurring to develop effective diagnostic methods and therapeutics. A study published in 2016 utilizes cell-free synthetic biology to develop a rapid sensor that is able to detect clinically relevant concentrations of Zika virus sequences in the plasmid of an infected monkey. The cell-free system utilizes a toe-hold switch sensor, which is a programmable riboregulator that contains a hairpin structure that prevents the translation of a desired gene by blocking off the ribosome binding site and start codon. However, when the trigger RNA binds to the switch sequence, it removes this hairpin structure, revealing the RBS and start codon and allowing for the translation of the gene. In this case, it is the LAC-Z gene, which when produced on a paper-based detector that they developed, it causes a color change from a yellow substrate to a purple product. To summarize, cell-free synthetic biology has a number of advantages over typical cell-based systems. You can manipulate the rates of transcription and translation due to the direct control of the open environment. There's the ability to synthesize and purify membrane proteins without difficulty. There's the ability to metabolically engineer enzymatic pathways without the concerns of cell growth and complexity, and there's no need to worry about the toxicity of the products. Although cell-free systems have a number of advantages and applications, much more research is needed in this field. Nonetheless, cell-free synthetic biology is becoming a very promising tool to overcome the worries and limitations of living cells. Thank you for watching.